Welcome to the 2009 Cell Signaling Technology in-house training tutorial for the study of stem cells. This tutorial contains 3 sections. Section 1 provides an overview of embryonic stem cells and ESC markers. This section also includes information regarding induced pluripotent stem cells, also referred to as iPS cells, and CST's new StemLite Pluripotency Kit. Section 2 covers information concerning stem cell differentiation along specific lineage pathways and corresponding lineage-specific markers. And finally, Section 3 covers epigenetic regulators and marks commonly found during embryonic development. What is an embryonic stem cell? Embryonic stem cells, also called ESCs or ES cells, are cells derived from the inner cell mass of a blastocyst. Looking at the figure on the right, a blastocyst is a structure formed early in embryogenesis, after fertilization, but before implantation into the uterine wall. A blastocyst contains approximately 70 to 100 cells and consists of an inner cell mass, also called the ICM, inside a cavity surrounded by a layer of cells called the trophoblast. Cells of the trophoblast later develop into the placenta and regulate implantation of the embryo. Cells of the inner cell mass are called embryonic stem cells and later develop into the new organism. Embryonic stem cells have 2 unique features. First, they are pluripotent. This means these cells can differentiate into all 3 primary germ layers, namely the ectoderm, mesoderm, and endoderm. Additional information regarding these germ layers will be presented in later slides. Secondly, embryonic stem cells have the ability to self-renew, which means they can replicate indefinitely in the undifferentiated state. Why are embryonic stem cells important? Embryonic stem cells are important for regenerative medicine. ESC pluripotency means that these cells can grow into any cell type in the body. Scientists study ESC pluripotency and differentiation with the hopes to create new healthy cells and/or organs in diseased patients. For example, new neurons could be created for patients with spinal cord injuries, or blood cells for patients with leukemia. Embryonic stem cells are also important for basic science research. ESCs are studied in basic research to increase our understanding of early-stage human development. The study of ESC differentiation along specific lineage pathways may increase our understanding of disease states and progression. Pluripotency at the molecular level. At the molecular level, the predominant signaling pathways involved in human ESC pluripotency and self-renewal are TGF-beta, which signals through SMAD2 and 3, and FGFR, which activates the MAPK and AKT pathways. The Wnt pathway also promotes pluripotency through activation of beta-catenin. Signaling through these pathways results in the expression and activation of 3 key transcription factors: OCT4, SOX2, and NANOG. These transcription factors activate gene expression of ESC-specific genes, regulate their own expression, and also serve as HESC markers. Other markers used to identify human ESCs are SSEA3 and 4, TRA160, and TRA181. Pluripotency and self-renewal are inhibited by the BMP4 pathway, which uses SMAD1, 5, and 8 to block transcription factor expression. The pathway shown is for human cells. Pluripotency in mice involves slightly different upstream signaling, primarily the LIF, leukemia inhibitory factor, and STAT3 pathway, but results in expression of the same key transcription factors. Confocal immunofluorescent analysis showing expression and nuclear localization of the transcription factors OCT4, SOX2, and NANOG in Entera2 cells. OCT4, SOX2, and NANOG antibodies have been labeled green. Actin filaments have been labeled red with DY554 phalloidin. Entera2 is a human embryonic carcinoma cell line that expresses the same markers as ESCs. Note the presence of green signal in these cells. HeLa cells are a non-pluripotent epithelial cell line used as a negative control and do not express these stem cell markers. Embryonic stem cell markers. Other markers commonly used to identify ESCs are TRA-160, TRA-181, and SSEA4, which are surface markers found on embryonic stem cells and germ cells. They are surface markers only and do not directly add to stemness or promote pluripotency. TRA-160 and TRA-181 are glycoproteins, and SSEA4 is a glycolipid. These antibodies recognize the carbohydrate group, not the proteins themselves per se. The functional role of TRA-160, TRA-181, and SSEA4 in stem cell biology is not clear. Confocal immunofluorescent analysis showing expression of TRA-160, TRA-181, and SSEA4 in Entera2 cells. Antibodies for TRA-160, TRA-181, and SSEA4 have been labeled green. Actin filaments have been labeled red with dy554 phalloidin. DNA has been labeled blue using the fluorescent dye DRAC5. Induced pluripotency, IPS cells. Induced pluripotent stem cells, IPS cells, or iPSCs, are a type of pluripotent stem cell derived from a non-pluripotent somatic cell by overexpression of a set of proteins. iPS cells have been shown to share many properties with ES cells, including epigenetic marks and the expression of stem cell genes. Two pivotal studies have shown that various combinations of key transcription factors have been used to obtain the induced pluripotent state in human somatic cells. In the first study, retroviral expression of OCT4, SOX2, c-MYC, and KLF4 in adult mouse fibroblasts caused the reprogramming of these cells into ES-like pluripotent cells. In the second study, a combination of OCT4, SOX2, NANOG, and LIN28 caused the reprogramming of human somatic cells into iPS cells. Since then, a multitude of studies have repeated these findings in various cell types. Other studies have focused on the necessity of individual transcription factors in somatic cell reprogramming. For instance, the activity of c-MYC and KLF4 can be replaced by the histone deacetylase inhibitor valproic acid. These studies have shown that OCT4 is the most important component for the creation of iPS cells because it is the only factor that cannot be replaced. The transcription factors responsible for somatic cell reprogramming function by promoting the expression of genes necessary for pluripotency and inhibiting lineage-specific genes. Because many of these genes control their own expression through autoregulatory mechanisms, the overexpressed proteins can bind the promoters of their endogenous counterparts, jump-starting and maintaining their own expression. There are many functional uses of iPS cells. First, iPS cells are very useful for drug screening applications. Researchers can screen compounds on more normal human cells and can better assess on-target and off-target drug effects at an early point in screening. In addition, compounds can be screened for their ability to drive or halt particular differentiation pathways, thus providing information on points of pharmaceutical intervention in particular disease states. iPS cells also provide the ability to generate cell model systems from patients with genetic diseases and allows scientists to follow disease progression as the iPS cells differentiate. A second use for iPS cells is that they eliminate ethical issues surrounding the use of embryonic stem cells. Human embryos would not be needed and as a consequence destroyed in order to obtain an original source of stem cells. And lastly, iPS cells can be used in personalized stem cell therapy. In the future, somatic cells would be taken from a patient and induced to become iPS cells. The iPS cells would be differentiated to create a cell type needed by that specific patient. For example, hepatocytes for a patient with liver failure. With this technology, there would be no need to worry about organ rejection or an immune response because the stem cells originated from that specific patient and have the same immune profile. New StemLite Pluripotency Kit. The new StemLite Pluripotency Kit contains antibodies against a selection of stem cell markers. This kit can be used to analyze the pluripotent status of human embryonic stem cells or iPS cells. Loss of marker expression indicates loss of pluripotency or differentiation of the culture. Retinoic acid treatment, 10 micromolar for 14 days, causes the differentiation of the embryonic carcinoma cell line Entera2 along the neuronal lineage. OCT4A and SOX2 antibodies have been labeled green. Actin filaments have been labeled red with dy554 phalloidin. Note loss of pluripotency markers green in the differentiated state. TRA160 and TRA181 antibodies have been labeled green. Actin filaments have been labeled red with dy554 phalloidin. Blue pseudocolor is DRAC5. Note loss of pluripotency markers green in the differentiated state. In the plus RA panels, RA treatment is causing the cells to differentiate from small round cells containing a lot of green signal into the larger elongated red cells located in the center of each panel. The red cells no longer contain the green stem cell markers. In the lower panels, neurofilament L is a marker for differentiated neurons, and the neurofilament L antibody is labeled green. No appearance of this marker in the differentiated state. Also note that the green signal is not present in the untreated pluripotent cells.